don't know how you met me, you don't know why you can't turn around and say goodbye. All you know is when I'm with you, I make you free and swim through your veins like a fish in the sea. I'm singing, follow me, everything is alright. I'll be the one to tuck you in at night. And if you want to leave, I can guarantee you won't find nobody else like me. I'm not worried about the ring you wear, 'cause as long as no one knows, then nobody can care. You're feeling guilty, and I'm well aware, but you don't look ashamed, and baby, I'm not scared. I'm singing, follow me, everything is alright. I'll be the one to tuck you in at night. And if you want to leave, I can guarantee you won't find nobody else like me.
So once we took our DNA samples from our fish, which was the tissue cutting, we put each tissue sample in a vial of Chelex. The Chelex basically makes the DNA more stable. So once we put the tissue sample in, we mashed it up and then centrifuged the vials so that the particulates would be separated from the clear fluid. Once we centrifuged the vials, we took the clear fluid off the top, which was our DNA, put them into separate containers, and then added primers. Our primers basically tag onto the DNA a section that we wanted, which was the CO1 gene. Once our PCR samples, our DNA samples, were ready to go through PCR, which is polymerase chain reaction, which this is after going into the thermocycler, we put the samples in, and basically polymerase chain reaction works in a way that the machine heats up the DNA enough to denature it, which basically unzips the strands of DNA. And then the primers that we put in previously tag on, which enables the DNA to be replicated once the machine cools back down. Samples come out of the PCR machine. We tried to test whether it worked or not, which basically tests if all of the steps in our protocol were successful, which there are a lot, so there's a very big chance that it wasn't successful. We know that our DNA is negatively charged, so in order for gel electrophoresis to work, the other side of the gel has to be positively charged. So we start by filling all of our wells in the bottom of the gel with our DNA samples, and then we turn on the charge, or the electrical current that runs through the gel, which is making the other side positive. In order to tell whether our sequences or our samples were successful or not, was that there would be an illuminated squiggly line kind of under our target line in each individual well in the gel. First gel from site A had 20% success, and we kind of expected this, or we didn't really expect more, because that was our first time running through the protocol, which is extremely difficult, and there are a lot of points in it where we could potentially mess up. Our site B gel, though, was much better. It had 80% success. Our successful samples are sent off to the University of Arizona. The University of Arizona is able to tell us the nucleotides and put it into a formal DNA sequence. Arizona sends back our sequences. They're in the formal ATCG format. We can actually put these into a program called FinchTV, which is an application online. Basically, we put the sequence in, and then any errors that might have occurred during the sequencing that Arizona did, we can manually fix. So once we fix all of our errors, we put the sequence into a system called the Barcode of Life database, and we submit it, and
So the advantages of DNA barcoding allows us to map speciation, to identify difficult species, which is sort of what you are all looking at. You're looking at species of Silversides that we can't identify because we're not good enough. But the DNA will allow us to do that. The research is important to us in Pensacola because it allows us to understand what animals we have around here, and to understand how those animals may migrate. So this is sort of the first part of a larger ongoing study. Right now, if we can establish what species are in Pensacola, then maybe later we can establish where their ranges are and what type of effects those animals have on the different ecosystems.
So for our hypothesis, we assumed that we would find Monidia berylina and Monidia peninsulae in our local waters, and we were technically right. A lot of our fish came back as they either double coated or they triple coated. They either coded for Monidia berylina and Monidia peninsulae, or they coded for Monidia berylina, Monidia peninsulae, and Monidia colii, which is an endangered species off the coast of Mexico.
So there are several potential explanations for the results that you got, especially with regards to the less popular species of Silversides that we had around here. Predominantly the berylina, and virtually every sample that you got came back as a positive identification for berylina. And then some of your samples came back as a potential for a different species that's common down in the Yucatan Peninsula, down in Mexico. From what we can understand, what Dr. Ebel was suspecting is that there may be multiple species that we have around here, one of which has probably not been sequenced, or if it has been sequenced, it hasn't been sequenced by a peer-reviewed researcher. And so what's happening is we're getting a false positive onto something that the BLAST software is saying, oh, it's probably this from Mexico, when it may just be a secondary local species around here that we just have never sequenced before. So what is it? Is it the berylina, or is it another one? Right now, we just have to say, don't know. That's a great thing in science, is to say, I just don't know, because then you can go on and ask more questions, and we could do it again next year. Species, and allow us to just that, but they're
please.
So Sienna Williams and her team, they did the Silverside DNA analysis, and this has become a very important, it will become a very important part of the diversity and abundance study, in that in the past we've grouped all Silversides into a single species, and in the future we'd hope to use their research to help identify the 3 or 4 different species of Silversides that are actually in our waters. We're going to do this probably finding those small morphological differences. In fact, Sea Grant agent Rick O'Connor came to the presentation the other day, and Sienna's research was presented, and he has already sent me some information about identifying these Silversides morphologically. Once we do that, instead of 1 species of Silverside, we could potentially have 3 or 4 species, which will impact our diversity and abundance numbers for the various sites that we're doing research on. So it's exciting. We're we're moving in the right direction. So thank you, Sienna, for your research.
So in the future, to further validate our results, future teams could possibly test more DNA sequences. They could look for more than just a CO1 gene. We could also either pick more sites from this area, or we could pick sites from different areas around the state or the country. Where all in all, our data won't be conclusive until we test outside of the CO1 gene. Happiness, hit
She hid in the wrong corners and she hid under bed. She killed it with kisses.
You have to sing at the same time. The dog days are over. The dog days are done. The horses are coming, so you better run. Run fast for your mother, run fast for Why are you doing that? Just stop. We're literally gonna do it again.
Densest student? That's a tough one for I have a really hard time picking out the smartest students and the densest students. It would probably be one of you. You're still recording.